It's my great pleasure and privilege to introduce our next speaker, academician, and also President Liao. Because even before he came back to Taiwan, he has enjoyed a great exposure in the news media, so that I really don't need to go very elaborate on introducing him. President Liao is expert, internationally renowned expert in metabolic engineering and also synthetic biology and, of course, the chemical biology. Before he came back, he was the chair of the Department of Chemical and Biomolecular Engineering at UCLA, and he was also the holder of the Ralph Paulson Foundation professorship. He has a long list of recognition and award. I probably cannot go through all of these within a few minutes, so I just highlight some of them. He was a recipient of the Presidential Green Chemistry Challenging Award in 2010, the White House Champion of Change for Innovations in Renewable Energy in 2012, the ENI Renewable Energy Prize bestowed by the President of Italy in 2013. And above all this, he was elected member of National Academy of Science in the US and the National Academy of Engineering. So it's without further delay, please join me to welcome the talk of President Liao.
Thank you, Professor Chen. It is truly my highest honor to be able to stand in front of everyone here to deliver the new academician's speech. Indeed, being elected as academic Sinica member is my highest honor. I truly appreciate this great honor and opportunity to speak in front of everyone. And we have heard two excellent talks this morning regarding various aspects in the biomedical research. I'm going to switch gear a little bit on a different philosophy, different line of research. Hopefully that will lead the science in a slightly different path. It is something that many people talk about called synthetic biology. And this line, this phrase has been coined a few years ago with different definitions. To me, synthetic means not natural. It's a synthetic fiber, that means not natural fiber. So when we are studying biology, most biologists will say that if it's not nature, if it's not in nature, it's not biology. So this is a little bit of oxymoron here. Okay? And I'll explain why. And I'm going to show with one particular aspect of synthetic biology. Synthetic biology has many, many definitions, many different applications. I'm going to focus on redesigning metabolism for the purpose of carbon management, okay, both on Earth and our human body. Okay, so you'll see a few things that I'm going to talk about. And because I know most of you are now working on this area, so I'm going to talk about several stories in overview format rather than going into the details, with the idea of introducing the concept. So why do we do this? In the past, we have been learning biology, sort of like we learn in kindergartens. You break things. You learn how things work by breaking it up. And that has accomplished a great deal. All we have done so far, learning all about the signal transduction pathways, all the different mechanisms, is because of this reductionist approach. We break things smaller and smaller, isolate from other interactions. Therefore, we learn things in precise manner. This is learning by breaking. Another way to do things is, after we learn enough, can we put them together to build new things? This is what I call learning by building. Maybe this is the time when you go to high school. You start building things that doesn't work. Okay? And we are probably in the junction between the transition of these two philosophies. We have learned a lot. We cure diseases. Diseases break our function, and we learn how to cure them. In the process, we learn how things work. Now, can we, based on what we have already learned, build new things, prevent diseases, or maybe cure diseases, or maybe solve some problems that human faces? All right, so a little bit background. I was trained as a chemical engineer. Okay. Chemical engineers are known to refine petroleum to make chemicals. After a while, I was kind of tired of doing the same thing. So in my graduate study, despite the fact that I have absolutely no biology background, I decided to learn about biology by applying chemical engineering principles to human systems. After all, human systems is a big chemical plant. So I did quite a bit of human studies on metabolisms, red blood cells, circulation, all that kind of thing. And then after a while, we switched to industrial problems. Can we shrink a chemical plant into a bacterial cell? We try to make different kinds of things using renewable resources. And this has been going on for 30 years. We were way ahead of time. Now this is, again, become one of the forefront of science as we speak. Now, moving forward, we're going to build really a chemical plant. We try to use plant to make chemicals that human beings cannot make at all. So all these are based on a few giants finding. We know that Louis Pasteur, about 1800, mid of 1800s, discovered that bacterial cells can actually conduct chemical reactions. So this was a big finding at that time. Okay? Life can conduct chemistry. In the next 20, 30 years, maybe 50 years, the details of this chemistry has been elucidated by a large number of people, including these two gentlemen, Emden Mayerhoff. They discovered, along with many other people, the details of glycolysis known as the EMP pathway. Okay, that becomes the foundation of every biochemistry textbook. Okay, you probably learned that in the second week of biochemistry. A few decades later, Hans Krebs discovered the TCA cycle, urea cycle, and about the same time, Melvin Kelvin at Berkeley find the CPB pathway that explain how cells, in particular plant cells, fix CO2 and make organic matters out of it. At that time, in my mind, this is the secret of life. This is the chemical secret of life. Unfortunately, this term, secret of life, was given to something else around 1950, which was DNA structure. After that, everybody jumped into DNA structure with lots of regulations and so on and so forth. And this line of research called metabolism was known as, quote unquote, solved. So everything else was, quote unquote, details. So no one paid any attention. Well, with a lot of exceptions. Now we are facing a different problem. We have a climate change problem. Why? Because we have lots of CO2 accumulated in the atmosphere, not just CO2, a lot of greenhouse gases, and with different potency. If we conduct business as usual, our temperature will rise above 3 degrees and maybe even higher. The CO2 level will exceed 600 ppm. What's a big deal? This is a very detrimental effect on Earth. It will cause irreplaceable damage. As we know that we have seen increasing frequency of climate-related disasters around the world. We have experienced a few in Taiwan in the past 10 years. If this trend continues, we will hit a temperature rise of more than 3 degrees. And the consensus is that we will have to limit the temperature increase at 2 degrees in the next 2-3 decades. And this is a pressing problem. And frankly, no one knows how to do this. And we all have to face the fact. So let's look at how to control the CO2 problem. Before the Industrial Revolution, this atmospheric CO2 is nicely balanced on land and on ocean, with photosynthesis taking up about 120 gigaton. The petagram carbon is the same as gigaton. Okay? I'm more used to gigaton as a unit. 120 goes photosynthesized, and 50 goes to the soil. I'm sorry, 60. About half of that decomposed right away back to atmosphere. And a similar idea happens in the ocean. So this is a nice homeostasis happened before Industrial Revolution. After Industrial Revolution, we begin to use fossil fuel. we produce about 6.3 gigatons. Where does that go? It has to go somewhere. It goes to the atmosphere. Okay? The homeostasis, the increase of photosynthesis wasn't high enough to absorb that. And that caused the annual increase of 3.2 gigatons. And we continue to do this. So the idea of solving this problem is, of course, hey, let's not use carbon. Let's use renewable electricity. This is great. We try to do the solar cells, wind, and so on and so forth. However, there's one problem. The solar cell and wind cannot, generated electricity cannot be stored. The only way to store electricity is using battery. Battery is good, but everyone has a cell phone, and when you want to make the most important phone call, it's out of battery. Why? Because energy density is too low. Energy density compared to liquid fuel, any liquid fuel, is about 1 to 50. The gasoline usually is about 40 times higher, and ethanol is not very high density. It has at least 25-fold higher. Okay, so this is the reason why we may still need to use liquid fuel for a long time, because we do not know how to solve this energy density problem. So then, how do we deal with this? Another idea is that let's make biofuel. So if you see that we take some of the photosynthesize the biomass and make that biofuel. So what you have done is you burn it right away. You go back to the atmosphere. It doesn't really make that much sense either. So we have to be a little bit more sophisticated. Of course, the analysis is not as simple as I said here. Okay? But largely speaking, we have to do something else. What is that something else? We will have to make sure that we increase the overall photosynthesis. There's no way around it. You cannot just take whatever that photosynthesizes. Maybe locally is good, maybe transiently is good, but ultimately we have to increase the overall photosynthesis on Earth, okay? So that we can take the photosynthesized, newly photosynthesized carbon, then use that to make biofuel. Then this kind of scenario will make sense. So then you will know that this is the reason why we need to do a lot of research on how to fix carbon, how to transform carbon to replace petroleum. So let's put a summary on everything we know about what I call the secret of life. Okay? We start from light reaction. This is on plant, on algae, and so on and so forth. Very complicated. We take light, convert light energy to ATP and electrons, and use that to fix CO2 using the Kelvin cycle that I just talked about. Of course, there are other pathways. And we use this cycle to make glucose. Some of them store in cellulose. When we need it, we convert cellulose back to glucose and then metabolize to make various kinds of metabolites for growth and for consumption and so on and so forth. One key point I want you to remember is that in all these pathways, there are two intermediates that's ultimately important. And this is in your Biochemistry 101, maybe the third week. Okay? One is called pyruvate, has 3 carbon. The other one is called acetyl-CoA, has 2 carbon. Okay? Between these two, we lost CO2, and this is the problem we're going to address later. So the first is that this is the composite idea of what happens in biosphere. Now we want to take the renewable resources, sunlight, CO2 outside to make these kind of products. Okay, the first question we ask is what to make. Okay, there are so many products, what to make. So a few years ago, almost exactly 10 years ago, we put together a pathway stealing from the bacteria the amino acid synthesis, the branch chain amino acid synthesis. Okay? And we found that we can extend some of the pathway, we can make longer chain alcohol. These are better than ethanol, so this has some immediate use. In particular, we extended the Leucine biosynthesis pathway to make isobutanol. So this process has been very successful, it's been commercialized. Unfortunately, in industrial scale, it didn't do very well for some other reasons. But this is not the focus of this talk. So after the 10 years, our lab and many other labs have demonstrated that we now can make just about everything we want. Okay, if you can write down the pathway, we can make everything you want using various microorganisms, maybe E. coli, yeast, or any other microorganisms. You can see the GC mass peak. The challenge is how to make them efficiently. That's number one. Number two is can you then use variety of feedstock? Up until now, most of the work are based on sugar, and we know sugar is food. We eat sugar every day. But we like to use non-food carbon source. So our lab has switched attention to engineer different organisms to use variety of different carbon sources, non-native carbon sources. One of the early ideas is that, hey, let's use CO2. Okay? Instead of using plant biomass, let's use a simplest photosynthetic cell. This is cyanobacteria. Okay? It's responsible to converting Earth environment from anaerobic environment to aerobic environment. Okay? So along the same line, we try to steal the intermediate to make various kind of fuel. Okay, the details of genetic engineering I'm not going to talk about, but we solve those problems. We have demonstrated that we can convert this cyanobacteria to make whatever you like, practically. Although efficiency is a problem, but in practice, we can make anything. Okay? We demonstrate that it is possible we can directly convert CO2 to any product with any chain length you like, if you can put together the pathway. The problem is still the efficiency. Okay? We have to focus on that. So the efficiency, for example, here we can make isobutanol at the milligram level less than 1 gram. So this is, overall efficiency is good, but in practice is not that practical. Okay, theoretically, this is the efficiency per liter, per hectare, per year basis is better than corn ethanol. Okay? But in order to achieve this, there are a lot of practical challenges. What are the practical challenges? One of them is engineering in nature. If you want to grow these photosynthetic organisms, you need to have a lot of photobioreactors. Okay, photobioreactors need to absorb light, so you need a lot of surface area to take sunlight. And instead of growing bacteria in a big vat, which is a 3-dimensional problem, we now change to a 2-dimensional problem, which increases the cost a lot. Okay, so this is a fundamental problem. It requires engineering solution, but so far, despite many decades of work, this problem has not been solved. So instead of solving this problem, let's go back to the drawing board. Do we have other ways to deal with this? This is the photosynthesis scheme from sunlight to biofuel. Okay? On every square meter, we get about 200 watts of sunlight. And we can calculate from sunlight to biomass. I didn't do this calculation. It was done by a group in Illinois. Theoretically, a C3 plant at a maximum theoretical yield from sunlight to biomass about 4.6%. And C4 plant is 6%. In practice, all less than 1%. Just remember this number. And for those of you who work on the photovoltaic cells, you'll laugh at these numbers. Photovoltaic cells, easy from Radio Shack. I don't know if Taiwan doesn't have Radio Shack. Whatever shop you buy a commercial photovoltaic cells, brag about the efficiency of 15%, far greater than this. The problem is that it stops at electricity. From electricity to something, chemistry is a big gap. Okay? So then the question is that can we combine the two strategies? Okay? Can we take advantage of the high efficiency of charge separation and electricity generation and somehow combine with biological activity to fix carbon? The answer is that if we can separate the light reactions with dark reactions, we may be able to do that. Okay? These light reactions and dark reactions are co-evolved, but there are so many cases they are separated. So if we can separate them, we can combine the photovoltaic generated electricity and use some chemistry to generate a chemical intermediate, for example, hydrogen, to drive Calvin cycle, and all we have to do is engineer this part of the microorganism to make biofuel. And if you calculate the theoretical efficiency, you can go to 9% instead of 5.7% using this type of scenario. And the most optimistic scenario in practice is probably get to close to 3%. This is pretty good. The problem is that now we use hydrogen, which is also create a lot of problems. For those of you who have a little bit of experience dealing with hydrogen, you know, first of all, you don't sleep at night. You worry about hydrogen explosion. Second, it has very low solubility. It doesn't go into water, so you need to agitate a lot. That's energy. We are trying to make energy, but we spend more energy, so that's not good. So the solution to that is, instead of making hydrogen, we make formic acid. Okay, formic acid can also be made using electrolysis type of reaction with the appropriate catalyst. For those of you who are chemists, you know this is an interesting problem to work on, and it's still in its infancy because no one is interested in making formic acid up until now. There's just no use for that. Okay? Now, formic acid can also drive Calvin cycle. And there are quite a few microorganisms called little autotrophy, okay, can utilize this kind of inorganic compound to drive Calvin cycle. So we take one of them called Rostonia eutropha. We figure out how to engineer those organisms and put in the pathway. We demonstrate it can make formic acid, it can take formic acid and CO2 and make all this biofuel. Okay. Long story short, we can assemble a device where we integrate electrolysis with bioreactors. We put in electricity either from sunlight or city grid. Then we can make gasoline substitute isobutanol from CO2 directly. So this accomplished the first demonstration of using CO2 and electricity to drive a non-photosynthesis. Okay, so this is the demonstration of this technology. And this, although that doesn't make very much sense to convert electricity to gasoline because electricity is probably more valuable. However, if you try to use renewable electricity, a lot of intermittent electricity cannot be stored, and that's a major problem. The wind power, the solar energy, it cannot be stored, as I mentioned earlier. The only way to store this energy is use battery, and which is very inefficient. If you can convert this energy to chemical energy, it can store forever. It can use in many different ways. So this becomes a very useful technology for storing energy, combining chemistry and biology. So moving forward, let's go back to this scenario. As I alluded to earlier, we can take CO2 or sugar to make whatever compound we want. However, in between, we have to go through this intermediate. Every cell using this type of idea. Go to pyruvate and go to acetyl-CoA, and acetyl-CoA is the precursor of everything. In between, we lose one carbon. There are 3 carbons here, there are 2 carbons here. So our maximum theoretical carbon yield is 66.6%. This is what I call the glass ceiling. It's there, but you don't see it. Okay? And everyone is suffering from that. And this is the fundamental reason why biochemical reactions cannot commercialize so efficiently today. So how do we deal with this? But what's the impact of this? First, let's look at the impact. This is the current scenario of converting glucose to ethanol. You lose 2 carbon. The maximum mass yield is 51%. If you say I want to make more advanced fuel, this is butanol, it's even worse. This is alkane, even worse. So changing the product doesn't change the efficiency. Okay? It's making it worse. We have to change the fundamental pathway. And this problem, because of 51% efficiency, ethanol costs about 80 cents per kilo. Glucose costs about 40 cents per kilo. You need 2 kilos of glucose to make 1 kilo of ethanol. This is where the government subsidy comes in play. This is why the farmers vote for Republicans. Now, the reason for this is between pyruvate to acetyl-CoA, we lose CO2 here. Can we design an alternative pathway to do that? So my lab came up with a very interesting idea, capitalizing on an enzyme called phosphokyrolase, which was isolated about 50 years ago. It cleaves a 6-carbon phosphate to 2 and 4. Another one is 5-carbon phosphate to cleave that to 2 and 3. So we can first take this 6-carbon, split into 2 and 4, and get 2 carbon out. The question is, what do you do with the remaining 4? So here the idea of chemical engineering comes in play. Chemical engineers know how to recycle things. Can we take dirty things, keep recycling? Okay, this is a product nobody wants. Can we recycle them, rearrange them back to the resource? So the idea is that can we design a pathway to rearrange the 4 carbon into 2 molecules of 6 carbon? And this turned out to be possible by a a little bit complicated scenario. I'm not going to go through all the carbon rearrangement cycle, but I guarantee this is also in Biochemistry 101. This is probably the 4th week. Okay, you learn something called the pentose phosphate pathway, and you hated it. Okay? As soon as you pass the exam, you forget everything about it, and you swear that you never learned it before. Okay? So these are all common pathways inside you and me. That means that the enzymes are very ubiquitous. So there are many choices that we can take from. Okay? You can follow the carbon flow and you can do that. The difference is that there are many cycles within cycles. Okay? Another way to look at this is that this is the more common way to look at the pathway. You see, this is double cycle. And this is probably the reason why that it escapes scientists for so long. Okay? So we don't know whether this type of cycle exists in nature or not. Probably it did, but no one found it. Maybe prove it. Okay? So what we did is that we construct this instead of breaking the cell to do this. Let's see, can we construct this pathway in the cell? Okay. The first we do is that we put together an in vitro system, demonstrate that we can do that in vitro, and we did do that. And we also showed that thermodynamically speaking, it is one way down here. There's no thermodynamic barrier. So this pathway is theoretically possible, actually better than a native EMP pathway. And we put together all these pathways in vitro, put them in vivo, and eventually put in the E. coli, optimizing a little bit. We demonstrate that we can achieve theoretical carbon yield without carbon loss. So this is the first demonstration that we can convert carbon to 2 carbon units to do whatever product you want in 100% carbon yield. Okay, so what is the impact? As I said, the traditional way of fermentation, probably the largest fermentation industry, is making alcohol. Okay, alcohol can be a fuel, can be a drink, whichever you like. Okay, so this is how you make the drink or the fuel. Take glucose to make alcohol. Now, if you want to make 3 moles of alcohol, you have to take 1.5 moles of glucose. But using our new pathway, this is called non-oxidative glycolysis. Okay, the traditional fermentation, the non-oxidative glycolysis, you only need 1 mole of glucose. But what's the penalty? You need additional electrons. Okay, comes in hydrogen. So the problem is that is hydrogen cheaper than half of the glucose? If hydrogen is cheaper, then you take this pathway. If sugar is cheaper, and if you don't care about the environment, you do the traditional way. Okay, so this is the balance. If you care about environment, you may want to pay a little bit more, or you might do a little bit more research to make it more efficient. Practically speaking, where do you get hydrogen? Okay. Oh, by the way, this type of idea, if you compare the traditional way with our non-oxidative glycolysis, it is essentially achieving what I call the hydrogen-assisted CO2 fixation. Okay, so we're using hydrogen to fix CO2 instead of using light directly. Another aspect is that where do you get hydrogen? Okay, today hydrogen, for those of you who are chemists, you know that the best way to get the hydrogen comes from methane. Okay, and the methane has enjoyed a lot of attention because the United States and many other countries found a lot of natural gas sources. After steam reforming, you can convert methane to hydrogen. Okay? If you use the hydrogen source, you produce some CO2. But overall speaking, you still do better than the traditional way. You still save half of the hydrogen. And this scenario is also can be understood as what we call the methane upgrading. We can add half of the carbon to methane to make ethanol. This type of reaction is not possible thermodynamically. So how can we achieve that? Because we couple a thermodynamically favorable fermentation pathway with a less favorable carbon-carbon arrangement pathway. Couple 2 together, we accomplish this. Speaking of methane, okay, this is the scenario in the United States. Produce a lot of different methane from natural gas, but the problem of methane is that if it has to be transported to the marketplace, so if you produce high enough methane, in terms of high enough rate, you can pipe it out to very low. If it's too low, then you cannot be used. So as a result, you just burn it on site. okay, called flaring. So this, below this line, is what people call the stranded methane. Stranded methane, the only way to deal with it is burn it. And that created lots of leakage and CO2. So this is a satellite vision on United States. You see the lights, New York, Boston area. You know, they're working very hard at night. All grad students are burning the midnight oil. And you see San Francisco partying, LA partying. What about here? This is North Dakota. What's going on in North Dakota at night? Those are flaring. The flaring, the CO2, the methane burned to CO2. Okay? So you can see the problem, how severe the problem is. So the idea is, can we utilize those low volume, low grade methane to make something? Methane utilization is has a long history, but it's actually not that easy. It requires a very high capital investment. Okay? For methane to syngas, or convert to methanol, the methanol to gasoline, also very, very large investment. That's difficult to do because of low volume and low grade. So ideally, we want to develop some biological way, because people criticize that biological ways are too slow and not efficient enough. In this case, we have a throughput, it requires low throughput. So it may be very good. The problem, again, there's no way to convert this biologically. Okay? A simple reaction, for example, 2 methanol, if you want to convert that to 1 ethanol. It seems to be a very simple reaction. Any high school student can write this overall reaction. Thermodynamically speaking, also very favorable, but there's no chemical way you can do that. Similarly, if you want to make even longer, 4 methanol to make 1 butanol. Thermodynamically very favorably, but not possible chemically. The reaction comes closest is this Gerber reaction that couples 1 and 3, not 1 and 1 or anything else. So the methanol to gasoline technology that developed by industry is a polymerization reaction that converts methanol to a longer chain non-specifically. So our challenge is, can we then design a novel pathway that nature hasn't derived? After all, this is our human problem. Nature doesn't have the responsibility to solve the problem for us. So we got back to this idea of non-oxidative glycolysis. We recognize that no matter you get from methanol or from C1 or CO2, it all goes through the sugar before metabolized to whatever pathway. And the same problem that we lose, we fix carbon, we lose carbon here. So if we can arrange this carbon to a way that we don't lose carbon, we may be able to save quite a bit. So again, use the same idea. We put together something called the methanol condensation cycle, combining the native UMP pathway and our non-oxidative glycolysis. Again, the cycle within cycles. We have demonstrated that this kind of cycle was able to convert methanol to C2 pathway. And we demonstrated that using C carbon rearrange tracing. By C1 labeling, if we use a 4 cycle, we can label 2 carbon in ethanol. Okay? If we cut out half of the cycle, then we get only 1 pass, only 1 label. So this experiment demonstrates that we indeed have a catalytic cycle. Okay, so we can put in 1 carbon and label both. Okay, why we do this? Because there have been a few papers in the literature claiming that they can do this, but at the end show that it was only a 1 pass, non-catalytic. So we had to demonstrate this catalytic reaction, and fortunately we were able to do that. Ultimately, we were able to convert methanol to ethanol with a reasonably high yield, and methanol to butanol also pretty good. And I have to remind you, at this level, we are doing in vitro study only. We're in the process of doing in vivo study. All right, so we covered that how to deal with this pathway, how to save CO2. Ultimately, we want to solve the problem of the native CO2 fixation. So what about this native CO2 fixation, the Kelvin cycle? This is what I call the secret, the chemical secret of life. The fundamental enzyme rubisco is the most abundant protein on Earth. Okay, on one hand, he said this is very important. On the other hand, he said this is very inefficient. That's why it's so abundant. Now, The problem of this pathway is that it requires lots of ATP to fix carbon. Okay, maybe good, maybe bad, but if we can create an additional pathway that's independent of Rubisco, we may be able to get a different highway. Okay? So our idea is design a novel pathway that fixes CO2 without using Rubisco. And we come up with this cycle. This is a reversal of glyoxylate shunt. Okay, for those of you who work on bacteria or plant, you probably know about this the glyoxylate shunt that goes the reverse way, but now we're trying to reverse that. And then if we arrange them appropriately, we can fix carbon here and fix carbon here. And this is the carbon fixation cycle. So we demonstrate this cycle in bacteria a little bit. Eventually, we put into Arabidopsis. And this is, I have to stress, this is the preliminary data. Okay? We are not 100% convinced that this is what we think is happening. So we knock out a CBB enzyme and demonstrate the Arabidopsis go very poorly. We put in our new pathway and was able to recover it, okay, to the growth. But this is still very, very early, and there are still a lot of controls that we have to do, and the results are not very stable. So at this point, I can only say this is an encouraging sign of success. This is not a final proof yet. All right. Up until now, we're solving problems on Earth. We have to look into ourselves. We also have carbon management problems. And the problem is the reverse. Okay? On Earth, we cannot generate enough energy. And here, we cannot consume enough energy. And you know the problem. I don't have to explain the importance of this problem. Again, the idea is that if you want to either fix carbon or burn carbon, up until now, most of the people working on regulation. Most of people drives, and some people drive in Taipei or in Los Angeles. If you live in these two cities, you know that traffic is a problem. If you want to solve the traffic problem, do you build more traffic light or you build more highways? Of course, you build more highways or subway, whatever. Regulation is like you build traffic lights. And you try to build more traffic lights or manipulate traffic lights to solve your problem. It helps, but not fundamentally. In order to fundamentally solve this kind of traffic problem, we have to build more pathways, subway or highway. So with that understanding, we decided to build additional pathway to burn CO2. Remember, this is the burning CO2, not fixing CO2. And just reverse of what we just talked about previously. We tried to reverse glyoxylate shunt. Now we just turn the glyoxylate shunt in the direction we want. This is a new CO2 burning cycle. We burn CO2 here. This is also similar to plants. In seed germination, uses a pathway to burn fatty acid, to use that to generate energy and various sources. So we had this idea almost 15 years ago. I wasn't able to convince any graduate student to try this because it was too risky. It could have no consequence. You know, cells are very resilient. They can resist whatever you do to them. So the most likely, no change or just dies. Second possibility, it actually can decrease fatty acid degradation because now if you put this pathway, if they go halfway, citrate goes out, and then citrate activate this, and then go on and so forth. A malonyl-CoA increase inhibits citrate, the fatty acid transport. So that's bad. Or it can go another halfway, go to this, go to gluconeogenesis. This is how plants do. Plants convert fatty acid to gluconeogenesis. This is not what we want to do either. There's only some possibility that it can increase the fatty acid degradation. So analyzing all these possibilities, I was having a great deal of hard time convincing any grad student to try this. Fortunately, about 10 years ago, I did have a very brave grad student decide to do this. And after quite a bit of trial, and this is the consequence, I'm going to skip a lot of details, we put this pathway, again, this is a synthetic idea, and this is by no means we can claim this is a therapeutic, but mice we can do that. We put this new pathway into mouse liver and fed the mouse with high-fat chow, and this is where you know what to eat for lunch, what not to eat for lunch. For the female mice, you can see that it gained weight dramatically. For the mice that we put in a new pathway, it retained a reasonably slim figure. And we, of course, we did a lot of analysis to show that the visceral fat is greatly reduced compared to the control, and this is actually increase in fatty acid the consumption. We do various metabolic labeling and show that it increase the respiration and so on and so forth. So finally, I'm going to leave you with this slide. Everything I said seems, if you can draw the pathway, it works. It's not true. We have a lot of failure. And another side of my lab is working on mathematical model. So we constructed a lot of mathematical models, understanding why it fails. It turns out it's a stability problem. Okay? If you start from something stable, you change the enzyme concentration, you very quickly can meet an unstable region. And this stability is dynamic instability. It's not exactly the same as the stability issue that biochemists are used to, not protein stability. This is a dynamic stability issue. So this is also a very rich area that interdisciplinary collaboration can occur. For those of you who are interested in systems biology, interested in bioinformatics, can work with people like myself and my students to understand what's going on, okay? To construct new pathways, new ideas, to move biology to a different level. To sum up what I said, the key point I'm going to leave to the audience is that nature evolved different mechanisms for the purpose of cellular survival, adaptation, and growth, not for humans to solve our own problems. We created our own problem. We cannot rely on nature to solve it. We have to do a little bit better, do our own homework to solve the problem we created. Fortunately, metabolic networks are very plastic. I demonstrate many ideas. You can change this plastic, but you have to do a lot of homework. You have to make sure that it's robust enough and solve all these issues in between. Finally, of course, I didn't do any of this work. This is people who actually do this homework, this work over the past 20 years. This is a composite picture of most of my students, previous students and current students. And I appreciate the funding from DOE, NSF, PAPAI, NIH, and the Kaiteki Institute in Japan. Thank you.
For this very stimulating talk. I think I would like to encourage graduate students to ask questions. So among the audience, you can graduate student raise your hand and ask questions.
Chemistry 101.
That is the tough part.
Yes.
Although you are not graduate student, we'll give you the privilege.
Sure.
Yeah, so I'm not a graduate student, but I try to behave like a graduate student. I really enjoyed the talk. For the new CO2 fixation pathway, the reversal of dioxylase cycle, when you put it in plants, the dioxys plants, did you put it into dioxylase dioxysomes, where those two enzymes are, or did you put it in chloroplasts?
We put in chloroplasts. We have several different type of promoters. We tried twice. Like I said, the result is still ongoing. We have one version expressed in the cytosol and transported in the chloroplast. And that's the one currently we're testing. And we're trying to do various tissue-specific promoters, and that's ongoing. This is a very complicated issue that we don't know what's going on. You know better than I do that plants is a lot more complicated. The silencing effects and this and that. So it's not easy. We're still working on it.
So if I could ask a follow-up question, in order to have the new pathway to work, do you have to knock out Rubisco first?
Okay. So we weaken Rubisco. That's why we show that the Rubisco mutant. There are three isozymes. We knock out two of them, and one's still there. And that one allowed it to grow a little bit. Okay, that's why we show that it grew, but the growth phenotype is dramatically reduced. So it didn't grow very well. So the idea is that if our new pathway can rescue this Rubisco phenotype, then it works. Probably. Okay, of course, there are other things. Thank you. Yes?
Thank you, President Liao. I'm the graduate student in Dr. Yizhang Chen's lab. I was wondering whether if we're working on the energy metabolism problem in neurodegenerative disease, can we further to modify the mechanism, like you mentioned before, to regulate the energy problem in neurodegenerative disease?
Definitely. Actually, you hit a very exciting area that I'm personally interested in pursuing. We should talk. I think one area that I didn't focus on is that you show everything I show is so nice, everything works. That's absolutely not true. 99% of things we try fail. Okay? And during the failure, you learn a lot more. So the kind of idea that you can do is that you perturb the system in a way that no one else did, and you learn a lot more different ideas. Up until now, in biology, the most common way of manipulation is knockouts. Okay? Now you can do knock-ins. Knock-in, not just the native function, but non-native function. Create something completely different and ask the question, what if? What happens? Create a hypothesis, and now you can link the traditional reductionist idea with systems idea. Okay? You create a different system, you ask a similar question. Okay? And I think that's a great area that deserves a lot of attention, and particularly in neurodegenerative disease. I think brain is one of the. Oh, by the way, for those of you who don't know, brain it consumes the most energy in our body. So you can see that we need a lot of energy supplied to brain. So that's ultimately important.
Okay, thank you very much.
That's great. And another graduate student?
Yes.
So basically, I'm interested in evolution. So since the synthetic pathway is not natural, so once you put those components into the cell, probably you will decrease the fitness. So how are you going to balance the production of the product you want and the basic growth for us?
That's an excellent question, and I will answer that very briefly. That's a blessing in disguise. Okay? Because as you know that we are doing this dramatic genetic engineering. A friend of mine called this is genetic engineering on steroids. Okay? We're doing a lot of cloning, a lot of genetic. We change cells dramatically. So the natural question is that safe? How does that impact the environment? Okay? How does that impact bioterrorism? Okay? All these questions that deserve a lot of attention, not just scientists. We have to be responsible, right? So that involves a lot of different aspects of regulation. Fortunately, whatever we created are not that stable. So we're not at that point yet. But someday, if it becomes very stable, we do have to worry about that the impact on biodiversity, impact on evolution. So you are asking the opposite question: how do I make it stable? Okay, and I will answer that by saying we only have to make it stable enough for us to do whatever we want. And the ideal case is that after this ideal confinement, if it releases to the environment, then it dies. That's perfect. Okay, we need to have a special greenhouse or a special reactor to grow these microorganisms. If it leaks out, it dies. Okay, and in fact, that's a very important area of research in synthetic biology is how to create such a system so that it can be completely confined. So you can see that there are many, many interesting questions to answer. Whatever question you ask, there's always a very rich research topic that you can create.
Wonderful. So we have 1 minute left, so open to graduate student.
No.
Honorary graduate student.
Yes. In your early speech, you mentioned that using synthetic biofuel may be one way to keep the CO2 under a certain level in the future. And you also mentioned that there is increase. There's almost no way to stop the increase of the CO2 in the near future, right? But I just want to mention that, or let you know that we have the technology in academia Sinica to make biochar from biomass in 1 minute.
Yep, yep.
So that we can use that to fix the carbon under the ground. And that may be one way to reverse the climate change.
That's a very good idea. And that goes back to what I mentioned earlier, is storing carbon on soil. Okay? If you look at the balancing chart in the beginning of my slides, let me quickly go to that here. What we are seeing is that you are taking photosynthesized compound and then store on soil. Okay, so that is good. As you can see that you reduce the decomposition. So that is definitely a very powerful technology. Yes. Okay. What I meant is that if you want to completely stop this, implementation is a problem. There's no way to say I have one technology and everybody on Earth is going to use mine. It's not going to work. So you can gradually do that. At the same time, we don't know what actually works. Okay? So my philosophy is that we don't pick winners. There are many parallel technology. We should all go forward. There's no way to declare winners in the beginning. Okay? From beginning on the research step to the implementation is a long way. We don't know. We don't even know at the end of the solution whether we have a mix of technology or we have one winning technology. Even that scenario, we don't know yet. So this is way too early to say which technology is going to win. So we just have to invest in many different ideas that make sense. Okay, so that's one of the ideas that makes sense.
So if I'm not mistaken, this is probably the first presentation you give in the capacity of the president of Academia Sinica.
If you talk about the Congress, that's the second one.
Okay. So please join me to give a round of applause to this very stimulating, and I think that under your leadership, we have a great future in terms of the area of renewable energy. Thank you so much.